Welcome back to BioMindscape. Now let's know what is a nephron. The structure of nephron. So nephron is a structure and functional unit of the kidney that is responsible for the urine formation. It has two main parts: the renal corpuscle, that is filtration unit, and renal tubule, that is processing unit. So the renal corpuscle, it is made up of glomerulus, a network of capillaries, and Bowman's capsule. So the blood enters through the afferent arteriole and leaves via the efferent arteriole. Here the unfiltration occurs to form the initial filtrate. Then renal tubule, this modify the filtrate and has several segments like PCT and DCT, that is proximal convoluted tubule, that maximize the reabsorption of water, glucose, amino acid and ions. And loop of Henle has a descending limb and ascending limb. So descending limb permeable to the water, whereas ascending limb is impermeable to the water. Here the reabsorbs salts. The ascending limb, it is impermeable to water and it reabsorbs the salt. That is important for the concentration of urine. And the distal convoluted tubule, it is selective reabsorptions. Secretions, it is regulated by the hormones. And the collecting duct, it final the concentration of the urine under the ADH control. Juxtaglomerulus apparatus, JGA, it formed by the macula densa and JG cells that regulate the blood pressure and filtration rate. So what is happening here? From glomerulus, the urine is passing to the Bowman's capsule, then PCT, then loop of Henle, then DCT, and then finally the collecting duct. So what is the renal corpuscle? It is a filtering head of the nephron which is always located in the renal cortex. It consists of the glomerulus, a tiny high pressure ball of capillaries where the blood filter actually occurs. The blood filtration occurs here. And Bowman's capsule, it is a double walled cup shaped chamber that surrounds the glomerulus and catches the filtrate. It is a cup shaped chamber that surrounds the glomerulus, the Bowman's capsule, and catches the filtered fluid. That is, it is doing the filtration work. Then arterioles, the afferent arteriole bring the blood into the corpuscles, while the efferent arteriole carries the filtrate blood away. Renal tubule, the tubule is long, winding duct that processes the filtrate into the urine through the reabsorption and secretion. So it is divided into several distinct segments like PCT, loop of Henle, DCT and collecting duct. So here what is happening? The first part of the tubule that is located near the corpuscle, it reabsorbs the majority of water, salt, glucose, amino acid. Whereas loop of Henle, a hairpin shaped loop that extends down in the renal medulla, it consists of the descending limb reabsorbing the water and ascending limb reabsorbing the sodium electrolytes. Then distal convoluted tubule, the final segment of the tubule which fine tune ion balance like calcium and potassium and regulate the urine pH. Then collecting duct, while technically not part of the single nephron, it receives the filtrate from multiple DCT, distal convoluted tubule, for final water reabsorption before emptying into the renal pelvis. Intercurrent mechanism is a physiological process where the fluid flow in opposite direction within the adjacent tubule, like the loop of Henle, vasa recta in the kidney, that create a high osmotic gradient. So this enable mammal to reabsorb the water and produce a high concentrated urine to conserve the water and maintain the electrolytic balance. So here the counter current multiplier, the loop of Henle create an osmotic gradient. The vasa recta, that is counter current exchange, it maintain the gradient and prevent it from washing out, often referred to the counter current multiplication or counter current exchange within the renal physiological studies. So here the kidney urine concentration, the loop of Henle in the medulla increases in intestinal osmoregularity, that is up to 12,000, which allow the water to be reabsorbed from the collecting duct. Then vasa recta function, the blood vessel parallel to the loop of Henle that transport the reabsorbed water and solute back to the bloodstream. Then seabird use the counter current. The salt gland regulated in the seabird. Seabird use a counter current mechanism to concentrate salt in the salt glands, allowing them to drink the seawater. How this mechanism works: the filtrate moves down the descending limb and up to the ascending limb of loop of Henle. The ascending limb actively pumps the NaCl into the surrounding medulla, making it hypertonic. Due to the high osmotic pressure, high NaCl in the medulla, water flows out of the collecting duct to the concentrated urine, and the blood enter the medulla and lose the solute but gains them back in it as it exits, so preventing the gradient from the dissipating. So, for knowing such concepts, follow BioMindscape.